All right, welcome back to part three in our metabolism chapter. We will go ahead and dive right in with glycogenolysis. So, when blood glucose drops, like between meals or during intense exercise, the body can't just make new glucose instantly. Instead, it turns to stored carbohydrate reserves in the form of glycogen. And this process of breaking down glycogen back into glucose is referred to as glycogenolysis. Now, glycogen is a highly branched polymer of glucose stored mainly in the liver and muscle tissue. And the enzyme phosphorylase breaks down those glucose units off the chain one by one, or it breaks down the glycogen into those glucose units one by one, releasing them as glucose 1 phosphate. And this step requires the coenzyme PLP, which is derived from vitamin B6. Now, that phosphate group is later rearranged into glucose 6 phosphate, which can either enter glycolysis for ATP production, or it could be dephosphorylated to free glucose, or dephosphorylated to free glucose and released into the bloodstream. So, entering glycolysis for ATP production, we'll see particularly in the muscle with muscle glycogen. The dephosphorylation to free glucose and released into the bloodstream would be where we would see liver glycogen, or the process that liver glycogen would undergo. So, liver glycogen maintains blood glucose levels, keeping the brain and other tissues supplied with energy. Now, muscle glycogen, on the other hand, is used locally. So it fuels the muscle fibers themselves during the contraction process, and especially early in exercise before fat oxidation increases. So only the liver, contains the enzyme glucose 6 phosphatase, which allows it to release free glucose into circulation. So, like we just talked about, glycogenolysis happens in both the liver and skeletal muscle. But again, the purpose and outcome are going to be different. So, the liver's job is to help regulate blood glucose for the entire body. The muscle's job is to fuel its own activity, so it doesn't really share. And so, when we look at liver glycogenolysis, we see that when blood glucose levels fall, such as during an overnight fast or between meals, the liver activates glycogenolysis. So the liver breaks down glycogen into glucose 6 phosphate and then uses the enzyme glucose 6 phosphatase to remove the phosphate group, and that allows free glucose to then be released into the bloodstream, supplying other tissues, particularly the brain and red blood cells, which depend heavily on glucose. And so this is a systemic energy response. The liver almost acts as like the glucose bank for the rest of the body. Now, muscle glycogenolysis, in contrast, we see muscle tissue breaks down glycogen for its own personal use. The muscle cells lack that glucose 6 phosphatase, so they can't convert that glucose 6 phosphate into free glucose, which would allow it to be released into the blood. Instead, that glucose 6 phosphate enters glycolysis right inside the muscle cell to make ATP locally for contraction. And so when we train, whether we're sprinting, lifting, this local breakdown of glycogen directly fuels the muscle fibers doing the work. So the two main glycogen storage sites are the skeletal muscle and the liver. And together, the healthy adult stores only about 415 to 425 grams of total glycogen, which is only about 11 and a half ounces of stored carbohydrate. So think about how big a 10-ounce steak is, and that's almost our complete carbohydrate stores throughout our entire body. So it's a relatively small energy reserve when we consider how dependent our body is on glucose during high intensity activity. And so when we break down the two sites, we see skeletal muscle is going to uh, uh, be the primary storage site for glycogen. We have about 325 grams of glycogen stored throughout our skeletal muscle system, which makes sense when you think about the total area of our skeletal muscles. Think about our arms all the way down to our legs versus the liver, which is a fairly large organ, but only one uh, organ. So this glycogen stored in skeletal muscle is reserved only for the muscle's own energy needs during contraction. So it, again, can't be released into the bloodstream. Now, the liver is going to be the other primary storage site of glycogen, holding about 90 to 100 grams of glycogen, and the liver's glycogen can be broken down and released as glucose to help maintain blood sugar levels, especially during fasting or long duration exercise. Now, once glycogen stores in the liver and muscle begin to drop, so again, during things like prolonged fasting or endurance exercises, once we see the glycogen stores start to become depleted, the body still needs to maintain blood glucose for the brain, red blood cells, and nervous system, but we are losing our reserve stores. So this is where gluconeogenesis comes in, which can essentially be translated or literally means making new glucose, right? So gluconeogenesis is the synthesis of glucose from non-carbohydrate sources. And this process primarily occurs in the liver and to a lesser extent the kidneys during a prolonged fast, but it's essentially the reverse of glycolysis. So it uses many of the same enzymes, but in the opposite direction, and it requires ATP to drive this process. Now, the body uses several non carbohydrate precursors, including glycerol, which is, remember that triglyceride backbone that the three fatty acids are attached to. You also have lactate from anaerobic metabolism. Remember going back to the Cori cycle that we talked about. You also have pyruvate, which is that intermediate from carbohydrate metabolism, and glucogenic amino acids from protein breakdown, especially during fasting and starvation. And so while your triglycerides or fats contribute glycerol, most usable glucose actually comes from protein. So yes, we can break down those fats in order to increase our glucose production, but majority of the time, if we are breaking down one of the other main macronutrients, it's going to be protein, specifically that the structural components of muscle and tissue that are going to be broken down into their amino acids for fuel production. So we generally see, say, a loss of muscle or muscle being broken down to drive gluconeogenesis. Now, gluconeogenesis is energy costly. So, like I said, it requires ATP from fat oxidation to power glucosynthesis. Now, this helps explain during prolonged fasting how fat and protein are both broken down. So, fat provides energy, and protein provides the carbon skeletons in order to rebuild glucose. Now, the body has multiple energy reserves, but triglycerides, which are the storage form of fat in the body, or the primary storage form of fat in the body, are by far going to be the largest and most energy dense. So, triglycerides provide more than twice the energy per gram compared to carbohydrates or protein. And this makes fat the primary long term energy storage form in the body. Now, the body can extract energy from dietary fat or from stored fat in adipose tissue. The body can also store much more energy as fat than as glycogen, since fat is compact and water free. And the body can also mobilize fat stores when glucose levels fall or during prolonged exercise or fasting. Now, fat is slow to mobilize, but incredibly efficient. And this is why it fuels low intensity, long duration activity, because it's so slow to actually mobilize. But once it does, you are able to produce so much ATP. And so when we look at the chart, we see that we can look at the macronutrient fuel content, and the majority of our fuel is actually stored as fat. About 78% of our energy storage is fat, whereas protein we have about 21%, because we have to take into account the skeletal muscle that could be broken down and used as energy if need be. And in contrast, you see carbohydrate stores are very limited and only make up a very small percentage of our stored energy reserves. Now, since carbohydrates are going to be our primary fuel source, but our stores are so limited, we see that when glucose levels drop, the body turns to stored fat for energy. And this process begins inside fat cells and is going to be controlled by a key enzyme referred to as your hormone-sensitive lipase. Now, fat cells contain hormone-sensitive lipase, which initiates lipolysis, which is going to be the breakdown of triglycerides into 3 fatty acids and 1 glycerol molecule. And this enzyme removes the fatty acids from that glycerol backbone, allowing them to be released and transported through the bloodstream. Now, when we look at the activation, we see that hormone-sensitive lipase is activated when blood sugar and insulin levels are low, again, such as during exercise or fast. And so this ensures that fat is only broken down when glucose is scarce, and this allows the body to prioritize carbohydrates whenever available. Now, once lipolysis occurs, we see fatty acids enter the bloodstream and attached to albumin, which is a water-soluble protein that carries them to tissues like skeletal muscle, maybe your heart, liver, for energy production. The glycerol portion travels to the liver, where it then can be converted into pyruvate or glucose through gluconeogenesis. Now, before a fatty acid can be metabolized for energy, it must first be activated in the cytoplasm. And this activation happens when the fatty acid binds to coenzyme A to form fatty acyl CoA. And this prep step is going to cost 2 ATP. This is that initial investment phase before entering the mitochondria. Now, the activated fatty acyl CoA then crosses the outer and inner mitochondrial membranes where beta oxidation occurs. Now, beta oxidation is the chemical pathway that catabolizes fatty acids within the mitochondria. So it breaks down the long carbon chain. Remember, we talked about the long chain fatty acids. They could be 16, 18, 14 carbons linked together. So it breaks down this long carbon chain 2 carbons at a time. And when we break these long chains into smaller 2 carbon molecules, we form acetyl-CoA with each cycle. So we are essentially cleaving or cutting or breaking apart the 2 carbon segment from the rest of the chain and joins with coenzyme A to form acetyl-CoA. And each cleavage is going to also produce one NADH and one FADH molecule, which carry those hydrogen or hydrogen ions or high energy electrons to the electron transport chain. And each NADH, FADH molecule from beta oxidation together will yield approximately 4 ATP once they reach the electron transport chain. And remember, we're getting 4 ATP essentially every time we break 2 carbons off of that long carbon chain. So this is a process that's going to occur multiple times whenever we are breaking down that long fatty acid. And so if you take palmitic acid as a classic example, because that's the primary fatty acid that we will burn, and it's a 16 carbon fatty acid, we see activating the fatty acid is going to cost an initial investment of 2 ATP. But then with our beta oxidation cycles, we have 7 rounds because remember, each round is removing a 2 carbon segment. So each round produces 1 NADH and 1 FADH molecule. And if we have 7 rounds, that makes 7 NADH, 7 FADH molecules, which gives or yields about 28 ATP just from breaking down that long chain fatty acid. Now, we also see that we have 8 acetyl-CoA molecules that are produced from breaking down this fatty acid, and each acetyl-CoA enters the citric acid cycle, which is going to generate approximately 80 ATP whenever we consider the substrate level phosphorylation, getting 1 ATP from each cycle, and then also the electron transport chain as we produce more NADH and FADH molecules during this oxidative metabolism. And so when you add everything together, you have about 80 ATP coming from the citric acid cycle. You have 28 ATP from beta oxidation, where we broke down that long chain into those 2 carbon molecules. And that would give us a total of about 108 ATP. But remember, we invested 2 ATP at the beginning, so we have a net total of approximately 106 ATP from 116 carbon fatty acid molecule. And so this just demonstrates how fat is extremely energy dense, because each fatty acid can go through this process multiple times, producing enormous amounts of ATP compared to carbohydrates. So longer fatty acid chains yield even more ATP. We just used an example of 16. We've talked about 18. 14 would produce a little less. And so this explains why fat is the primary energy storage the primary energy source during prolonged low intensity activity, and also why it takes oxygen dependent pathways to fully metabolize fats. Now, human cells can't use fatty acids to make new glucose. Only the glycerol backbone of triglycerides can be converted into glucose, and again, that process occurs in the liver through gluconeogenesis. And so maintaining some glucose availability is vital, because not all tissues can use fat effectively, such as the brain, nervous system. Red blood cells rely very heavily, potentially even exclusively or nearly exclusively, on glucose as their main energy source. And so red blood cells, for example, lack mitochondria, so they can't oxidize fatty acids or even run the citric acid cycle. So we also see the brain prefers glucose because fatty acids can't cross the blood-brain barrier efficiently. So after eating, cells use glucose first to regulate blood sugar and meet immediate energy demands. And once glucose levels return to normal and insulin drops, the body switches to burning fat for energy. And this transition between glucose use and fat oxidation is known as the Randle cycle, or the glucose fatty acid cycle. Now, lipolysis and beta oxidation are inhibited by insulin and high ATP levels. This signals that energy and glucose are sufficient, right? So on the flip side, we see lipolysis and beta oxidation will be activated by glucagon, which is a hormone that rises when blood sugar is low. And this ensures fat breakdown is only triggered when needed, like during fasting, prolonged exercise, or potentially between meals. So when energy and glucose are abundant, insulin and citrate, which is a byproduct of carbohydrate metabolism, activate fatty acid synthesis. So this is the process of converting extra carbs into stored fat. Conversely, when glucagon inhibits the synthesis of fatty acids, from glucose, or extra carbs, and shifts the balance toward fat oxidation. So insulin citrate activate fatty acid synthesis from extra carbs, and glucagon inhibits this synthesis, shifting the balance back towards fat oxidation, burning fat, or using it as fuel. Now, when glucose is unavailable, again, such as during periods of starvation, fasting, maybe you're consuming a very low carbohydrate diet, the body must find an alternative fuel source. And so the liver begins converting acetyl-CoA from fatty acid oxidation into ketone bodies in a process called ketogenesis. And this allows most cells, particularly the muscle and brain cells or tissue, to continue producing ATP when glucose is scarce. And there are three main ketone bodies, which are acetoacetate, beta hydroxybutyrate, and acetone. Now, these are produced in the liver from acetyl-CoA and then released into the bloodstream to be used by other tissues for energy. And so it is important to note that liver cells can't use ketones because they lack the necessary enzyme to reconvert the acetyl-CoA into usable energy for the liver. Now, acetoacetate and beta hydroxybutyrate are both usable fuels, and they can enter other cells like the muscle and brain and be converted back into acetyl-CoA, thus feeding directly into the citric acid cycle for ATP production. On the flip side, acetone is a byproduct more so of the acetoacetate breakdown, and it is going to be produced in relatively small amounts. It's not metabolized, and it's exhaled through the lungs, and this is what gives the maybe fruity odor that individuals may have on their breath if someone is in ketosis. Now, in the early stage of carbohydrate restriction, acetone levels rise and can be measured in an individual's breath as a sign of ketosis. And as the body adapts to long-term ketosis, it becomes more efficient. So the liver produces fewer waste ketones. Peripheral tissues like the muscle and the brain increase their uptake efficiency, and acetone levels in the breath decrease even though fat oxidation continues. So a ketogenic diet is defined as a high-fat, moderate to low protein, and very low carbohydrate diet or eating plan. And so the goal is to shift the body's metabolism from using glucose as its main energy source to relying on fat and ketone bodies. So for most people, consuming less than 50 grams of carbohydrates per day is enough to trigger this ketogenic state. And at this point, the liver glycogen will be depleted and the liver begins converting fatty acids into ketone bodies for fuel. So ketosis is a controlled adaptive metabolic state where the liver produces moderate amounts of ketones to meet energy demands. Ketoacidosis, however, is a dangerous, uncontrolled condition that occurs primarily in people with diabetes, especially type 1 uh, diabetes. And it results from a lack of insulin, which causes excess acetoacetate and beta hydroxybutyrate to accumulate in the blood. And this lowers blood pH, leading to acidosis. So, some key warning signs here would include excessive thirst, frequent urination, blood glucose greater than 250 milligrams per deciliter, and that distinctive fruity odor on the individual's breath. And so, if untreated, ketoacidosis can progress to where an individual could possibly enter a coma, or we could even see death as a potential consequence. Now, when done safely and properly monitored, the ketogenic diet can reduce hunger by stabilizing blood sugar and influencing your appetite hormones. It can lower insulin levels, which allows for greater lipolysis or fat breakdown, and thus fat mass reduction. And it can improve insulin sensitivity and blood lipid profiles in certain individuals. So, clinically, it has been used as a therapeutic tool for conditions like obesity or metabolic syndrome, type 2 diabetes under strict supervision, epilepsy, Alzheimer's, and some cancers are even under investigation as well. So, ketosis is a normal adaptive process. It's the body's way of maintaining energy when carbohydrates are scarce. But ketoacidosis, not the same thing. It's dangerous overproduction of ketones due to insulin absence. So, ketogenic diets can be beneficial, but context and regulation are essential if you are potentially looking at switching to one of these diets, especially for individuals with diabetes or metabolic conditions. Now, like we talked about with our proteins chapter, under normal conditions, cells primarily use amino acids to build essential proteins, whether it's structural or certain enzymes or regulatory proteins. And once those needs have been met, any excess amino acids can be catabolized for ATP production, or they can be converted into glucose or fatty acids for storage for later use. Now, it's important to remember cells always use some amino acids, like very, very little for ATP, but glucose and fatty acids are going to be the primary energy sources. Now, you can think of protein as more of a backup fuel. So it's used in periods of starvation or very long fasts or intense training where intense prolonged training, I should say, where we've depleted a lot of our other fuels. So to use amino acids for energy, cells must first remove the nitrogen-containing group or the amine group, and this nitrogen removal allows the remaining carbon skeleton to enter metabolic pathways like glycolysis, like the citric acid cycle, or gluconeogenesis. However, the removal of nitrogen must be handled carefully, because when removed, we see it will form ammonia or NH3, which can be toxic at high levels. Now, both deamination and transamination require the coenzyme PLP. Now, if you don't remember deamination and transamination, remember deamination is the direct removal of that amine group from the amino acid, which produces that ammonia, which is later converted to urea and then safely excreted. Transamination, instead of discarding the amino or amine group, you have the cell that transfers it to another molecule. So this process helps new amino acids form, or in this case, since we're talking about metabolism, it helps form intermediates for metabolism. And so transamination, think more of nitrogen recycling, where deamination you are discarding or repurposing in transamination and immediately eliminating in deamination. Now, like I said, both deamination and transamination reactions require PLP, and PLP is derived from vitamin B6, and so this makes vitamin B6 essential for amino acid metabolism and energy production. And so a vitamin B6 deficiency can impair protein metabolism, reduce energy availability from amino acids, and disrupt nitrogen balance. Now, once the nitrogen group has been removed, the remaining carbon skeleton of the amino acid can enter the citric acid cycle. And depending on the amino acid or the carbon skeleton, we can see that there are different entry points for amino acid metabolism. So you have your glucogenic amino acids, which can be converted to pyruvate, and thus we would see the same process as that one molecule of glucose that's broken down into pyruvate before being converted to acetyl-CoA and entering the citric acid cycle. So these amino acids are important during either fasting or carbohydrate restriction, and this is when glucose must be synthesized in order to maintain blood sugar and brain function. And so some common examples, you have alanine, you have cysteine, threonine are common glutogenic amino acids. Then you have your ketogenic amino acids, which can be broken down into acetyl-CoA, acetoacetate. What we see is that these products can't be used in any way to make, say, glucose. Instead, they feed into the citric acid cycle for ATP production, or are converted into ketones during fasting or your low carb states. And so the main ketogenic amino acids are leucine, lysine. Both are purely ketogenic. Others, such as tyrosine, isoleucine, are going to be both ketogenic and glucogenic, or depending on the situation, can act either ketogenic or glucogenic. And so you have amino acids that are both glucogenic and ketogenic, and this means they can be converted or act as glucose or ketones depending on the body's needs. So, for example, isoleucine, threonine, phenylalanine, tyrosine, tryptophan, all of these amino acids, their carbon skeletons can act as glucogenic or ketogenic amino acids. And this metabolic flexibility is useful during prolonged fasting because it allows cells to adjust fuel use dynamically. Now, the total amount of ATP produced from an amino acid depends entirely on where its carbon skeleton enters the citric acid cycle. So, as shown in the diagram, you have that amine group that is removed, and you have the remaining skeleton that can enter either as pyruvate, which is feeding into that glycolysis pathway, if you want to consider it acting more as like a glucose molecule. And then you also have your citric acid cycle intermediates, which are going to be used to directly fuel oxidative metabolism, entering at some stage in the citric acid cycle. And so the closer the entry point to the start of the energy path, the more ATP will be produced. And so your glucogenic amino acids that are converted into pyruvate are going to produce more ATP than say your amino acids that would enter the citric acid cycle halfway through. And so even though amino acids can produce ATP, we see they are much less efficient energy sources than either glucose or fatty acids. So amino acid catabolism generally yields much less energy than carbohydrates or fats because, one, the energy cost of removing nitrogen, and two, the fact that amino acids enter the citric acid cycle potentially midway through the pathway. And so the body typically preserves amino acids for protein synthesis only and relies on them only for ATP production when absolutely necessary. Again, during periods of starvation or very prolonged exercise, we're talking probably 90 minutes plus. Now, the body stores excess energy whenever fuel intake exceeds immediate energy needs, and these stored forms act as reserves or storage sites to draw from whenever fasting or exercise occurs. And the main storage sites are going to be the liver, skeletal muscle, and adipose tissue. And they store energy in the form of glycogen or triglycerides. So when we look at the numbers, you have liver glycogen, which only stores about 300, 400 calories. And we see our liver glycogen is primarily used to maintain blood glucose during fasting. And if you're wondering where the 300 to 400 calories came from, remember we talked about how liver glycogen is about 90 to 100 grams. Just multiply that by 4. And then you have your muscle glycogen, same thing, multiplying those values by 4, we get about 12 to 1600 calories. But these stores, the muscle glycogen stores, are used locally within the muscle during activity, right? It can't leave the muscle. And then you have your adipose tissue, which has approximately 80 to 100,000 calories of stored energy, making this by far the largest energy reserve. Now, when energy is needed, the body follows a specific order of use. So we have blood glucose, which is, I guess you could say, that first line of fuel, glycogen stores, which is a short-term energy backup once we see glucose levels start to drop. And then you have your stored fats or triglycerides that are going to be your long-term energy source that has a very high capacity, but it takes a while to kick on. And then you have your body proteins, which are only broken down as a last resort, such as during starvation or the prolonged endurance events. And so this hierarchy helps preserve lean tissue and maintain metabolic balance. Now, fat provides more than double the energy per gram compared to either carbohydrates or fat. Remember, carbohydrates and proteins, we get 4 kilocalories per gram, whereas fats, we get 9 kilocalories per gram. And even a lean individual still carries tens of thousands of kilocalories worth of fat energy, which is going to be enough to sustain potentially weeks of fasting, or at that point, I'd say starvation. Now, the trade-off is that fat can't support high-intensity exercise alone, since it's metabolized more slowly than carbohydrates. So we still need our carbohydrates as our fuel source for high-intensity exercise. Now, as we said on the last slide, when a person consumes more macronutrients than needed for immediate energy, the body has to store or convert the excess. And so depending on the macronutrient, this storage can take many different forms. You have glucose, which can be stored as glycogen or converted to fat. You have fat, which will be stored as triglycerides. You have amino acids that can be converted to glucose or fat when we consume an excess amount. So glycogenesis is going to be the process of linking glucose units together to form glycogen. And this occurs mainly in the liver and skeletal muscles, which is where we see most of our glycogen stores. Now, specific enzymes connect individual glucose molecules into long branch chains for efficient storage. However, glycogen storage is limited. Right? So once glycogen stores are full, any excess glucose will be converted into fatty acids for long-term energy storage. And this is where we'll talk about lipogenesis, which is the synthesis of fatty acids from acetyl-CoA units. So you have those 2-carbon acetyl units that are joined together to form a hydrocarbon chain of a fatty acid, so essentially the opposite of beta oxidation. And so when glycogen stores are full and ATP levels are adequate, we see glucose converted to acetyl-CoA, converted to fatty acids, which are then attached to form triglycerides and stored in our fat tissues or adipocytes. Now, this conversion ensures that no excess energy goes to waste, but it also explains why overeating carbs or even fats can lead to fat accumulation. Now, on the flip side, when energy is scarce, these pathways generally reverse direction and mobilize glycogen and triglycerides for ATP production. And then gluconeogenesis, we see we can break down our glucogenic amino acids to make glucose or our ketogenic amino acids as well, and we can see them converted to fat for storage. Now, as you can probably already tell by now, the body has an incredible ability to convert one macronutrient into another depending on energy status and dietary intake. And this table shows which nutrients can be converted to glucose, to amino acids, or to fat. And so understanding these conversions helps explain energy balance, muscle maintenance, and fat gain. So if the diet provides an excess amount of amino acids, your body doesn't need them to build proteins because you already have enough amino acids. So what happens to them? Well, your liver and kidneys can use the glucogenic amino acids to make glucose through gluconeogenesis. And then you have your ketogenic amino acids, which can be converted to acetyl-CoA and contribute to fat synthesis. Now, theoretically, all amino acids can yield fat because those glucogenic amino acids that are converted to glucose, if you have an excess amount of glucose, that glucose could then yield fat. So amino acids can all produce fat. Only the ones that are glucogenic can produce glucose or yield glucose. Now, glucose can yield fat. It can also yield non-essential amino acids, but only if nitrogen is available. So essentially, you'll have a carbon skeleton that nitrogen group could attach to. Now, triglycerides can't directly yield glucose except for that glycerol backbone, which can enter gluconeogenesis. And the fatty acid chains themselves can't form glucose because they break down to acetyl-CoA, which can't reverse back into pyruvate. And so triglycerides are pretty much just excellent at yielding fat. That's their only real role as far as energy storage. And then alcohol also will contribute to fat synthesis because it's broken down into acetyl-CoA, which we see can eventually yield fat. However, alcohol can't produce amino acids or glucose. Now, normally when excess calories are consumed, the body is going to store them or eventually use them for energy. But there's a third option where the body can actually waste some of the excess energy by converting it into heat rather than storing it as fat. And so this process is part of how the body maintains energy balance and can explain why some individuals potentially resist weight gain more effectively than others. So you have adaptive thermogenesis, which is the process of energy dissipation as heat instead of ATP. And so it primarily occurs in the mitochondria, especially in muscle tissue, and brown adipose tissue. And so instead of using or coupling food energy to ATP production, the mitochondria uncouple it, releasing energy as heat. And so this involves uncoupling proteins or UCPs that waste calories to keep the body temperature stable. And so these rates fluctuate with environmental temperature, with diet, and with hormones of an individual. So in cold environments, we see an increase in thermogenesis, and as a result, a raising basal metabolic rate. And then an overconsumption of calories also increases thermogenesis, while caloric restriction will reduce it. And so, in short, adaptive thermogenesis helps the body regulate body temperature and energy efficiency. Now, NEAT stands for non-exercise activity thermogenesis, which is that energy expenditure from spontaneous, everyday physical activities. So examples might include fidgeting, maintaining an upright posture instead of slouching, maybe shifting weight while you stand. It could include maybe any sort of light incidental movements throughout the day. And so NEAT is less efficient than structured exercise, meaning more energy is lost as heat and less energy is actually conserved for that movement. So depending on a person's habits and activity level, your non-exercise activity thermogenesis can waste anywhere from 100 to 700 calories per day. And this variability helps explain why two people with similar diets may gain or lose weight at different rates. So their unconscious activity levels differ potentially significantly. So together with adaptive thermogenesis and non-exercise activity thermogenesis, we see these are going to represent two very subtle but powerful ways the body can adjust to caloric intake. And so when energy intake rises, these systems can increase calorie waste to prevent any unwanted fat gain. And when intake drops, they can slow down to conserve energy, which is part of why long-term dieting becomes harder over time. Now, looking at the regulation of metabolism, when we eat, we see the body must decide whether the energy from the meal will be stored or burned. And this decision isn't going to be random, right? So it's largely controlled by hormones, which act as the body's chemical messengers. And these hormones are going to be secreted by specific organs such as the pancreas or adrenal glands or the thyroid, and they circulate through the bloodstream to reach target tissues like the muscle or the liver, adipose cells. And once there, they stimulate or suppress certain metabolic activities depending on the current energy state. So think of hormones as traffic directors for metabolism, so they can determine whether energy moves towards storage, like building glycogen, or towards catabolism, where we are breaking stored energy down to make ATP. And so the actions of these hormones shift depending on whether we're in a fed or fasted state. And this hormonal balance ensures the body maintains energy homeostasis even between meals or overnight when we are fasting for hopefully at least 8 hours. Now, the body's metabolic state can generally be divided into two main categories. You have the fed state, which is going to last from when you were eating to 4 hours after eating. This is where nutrients from the recent meal are being absorbed and processed, and energy is readily available. So the body prioritizes storage and anabolism. And then you have your fasted state, which is going to be anywhere 4 hours beyond eating your last meal. So the body has used up circulating glucose and begins mobilizing stored energy to maintain blood sugar and ATP production. So metabolism shifts to catabolism, where we're breaking down glycogen and triglycerides, maybe some amino acids as well. And so these two states create a metabolic rhythm that cycles throughout the day, where we're eating, storing, and then drawing from those reserves. Now, there are several key hormones that work in concert, I guess, to regulate these shifts. So you have insulin, which signals nutrient abundance. All right, so when blood glucose levels are very high, and so it promotes the storage of glucose and fat. You have glucagon, which is going to signal that energy is needed, which promotes glycogen breakdown. You have cortisol, which is a stress hormone that enhances glucose availability and promotes protein catabolism, or the breakdown of proteins. You have epinephrine, which is going to rapidly mobilize energy during the fight or flight response. And you also have the thyroid hormone, which regulates overall metabolic rate and is going to influence how fast energy is burned. So these hormones act dynamically, responding not only to an individual's nutrition, but also to stress, to activity, as well as environmental temperature.